Welcome to Tutorial Zone, where we simplify complex concepts. Before we dive deeper into this lesson, please do subscribe, share, like, leave a comment, and turn on your notification button so that every time I upload a new video, you should be notified. In this lesson, we are going to look at the anatomy and physiology of the cardiovascular system. We are going to introduce blood and we are going to look at the red blood cells. I am Dr. Said Piri, so let us begin. At the end of this lesson, you are expected to do the following. Define blood, state the functions of blood, state the physical characteristics of blood, describe the principal components of blood, explain the process of hematopoiesis, and describe the structure, functions, life cycle, and production of red blood cells. So we're going to start with the introduction to the cardiovascular system. The cardiovascular system is made up of three important components. It is made up of blood, it is made up of the heart, it is made up of blood vessels. So it is important to understand that the heart acts as a pump in this case. It pumps blood to body organs or tissues. And for this blood to reach the body organs or the tissues, it requires a conduit or a passage where the blood is going to pass for it to reach the body organs or the tissues. And therefore, these blood vessels act as conduits or passages by which blood reaches those body organs. You also have to understand what hematology is. So hematology is a branch of science which is focused with the study of blood. So under blood, in hematology, you can study things such as the components of blood, you can study the functions of blood, you can also look at the characteristics of blood. However, hematology does not just end on studying blood, its components and the functions. We get to also look at blood-forming tissues. So these tissues, we can talk about the red bone marrow. So we'll look at the red bone marrow in just some minutes from now. Hematology also does not end on blood-forming tissues. It also looks at the disorders that are associated with blood. So you can think of things such as, or disorders such as anemias, you can think of leukemias, and also bleeding disorders. So hematology looks at all these three components: blood, blood-forming tissues, and disorders associated with blood. So how do you define blood? Blood is defined as a liquid connective tissue consisting of cells surrounded by a liquid matrix, which is plasma. So in this case, what this means is that there are two parts of blood. We have the extracellular matrix, which is liquid. This one is our plasma. And in this plasma, we found cells suspended in the plasma itself. So two components, extracellular matrix, which is the liquid part, the plasma, and the cells are suspended within the plasma. We are now going to talk about the functions of blood. Blood has three important functions: transportation, regulation, and protection. When you're talking about the function of transportation, blood acts as a medium that transports several materials. For example, blood transports gases such as oxygen from the lungs to the tissues and carbon dioxide from the tissues to the lungs. It also helps in transportation of nutrients from the GIT to the body organs. It also helps in transportation of endocrine hormones from the endocrine glands to the site of action. It also helps in distribution of heat throughout the body, and also it transports waste materials for their removal. The second function of blood, which is regulation, blood plays a role in the regulation of pH. It plays a role in the regulation of body temperature, and it also plays a role in the regulation of homeostasis of body fluids. The third function of blood, which is protection. In this row, blood has got several components such as platelets. Platelets help in the protection against excessive blood loss. By forming a clot. Blood also helps in protection against infections. How does it help against protection of infections? Blood has got white blood cells that help in fighting off infectious agents in the body. It also has proteins such as antibodies that also assist or play a role in fighting off infections. So those are the three functions of blood: transportation, regulation, and protection. We are talking about the physical characteristics of blood. Number one, blood is denser and more viscous or thicker than water. The reason blood is denser and more viscous is because of the formed elements that are present in blood, which are the cells and the cell fragments, the white blood cells, the red blood cells, and the platelets. Then the other thing that makes blood thicker and more denser are the dissolved proteins that are present in plasma. Then the temperature of blood is about 38 degrees Celsius, which is about 100.4 Fahrenheit. So this temperature is about 1 degree Celsius higher than the oral and rectal temperature. The oral temperature is about 37 degrees Celsius, while the rectal temperature is about 37.5 degrees Celsius. The pH of blood is slightly alkaline. It has got a lot of bicarbonate in it, so it is slightly alkaline at the range of about 7.35 to 7.45, and the average being 7.4. The color varies with the oxygen content, in that when blood is more concentrated with oxygen, it is bright red in color, and then when it has got less oxygen, it is dark red in color. Blood also makes up about 20% of the extracellular fluid, which is the fluid on the outside of the cell. And then it makes up about 8% of the total body weight. So if someone is 70 kg, it means that 8% of this 70 kg is blood. Then the volume of blood differs between an adult male and an adult female, with adult males having more blood volume in comparison to adult females. For adult males, the blood volume is about 5 to 6 liters, while for an adult female it is about 4 to 5 liters. And this difference is because of the body mass. Males have got more body mass, have got more muscles in comparison to females, and therefore males require more blood to the muscles than females. And then other reasons, I'm going to mention them in the videos, especially when I'm talking about hematocrit. You're going to see why males have got more red blood cells in comparison to females. We can now look at the components of blood. Blood has got two major components: blood plasma and formed elements. Blood plasma makes about 55% of the blood volume, and this blood plasma is the watery extracellular matrix, which has dissolved substances, and these dissolved substances, they include nutrients, waste materials, electrolytes, proteins. The formed elements, these are the cellular components of blood, and the cellular components of blood include red blood cells and white blood cells, as well as platelets. Platelets, however, in comparison to the red blood cells and white blood cells, which are complete cells, platelets are simply just cellular fragments. By cellular fragments, what do I mean? They're just pieces that break off. They break off from a larger cell during production. I'm going to explain about that under hematopoiesis, and I'll also talk about it under platelets in the other video. These components of blood can best be visualized when we get a blood sample and then we put that blood sample in a small glass tube. Then we spin this glass tube and then allow it to settle. What will happen is that the red blood cells, which are very dense, are going to settle at the bottom. And then the plasma, which is less dense, is going to settle on top. In between the plasma and the red blood cells, we're going to have a combination of white blood cells and platelets. These two, they are less denser in comparison to red blood cells. They're not as heavy as red blood cells, but they are heavier than plasma. So because of that, they are going to settle on top of the red blood cells, but they are going to settle at the bottom of the plasma. And this layer of white blood cells and platelets is known as the Buffy coat. In terms of percentages, the red blood cell are going to make up 45%, while plasma will make up 55%. The Buffy coat, the white blood cells and the platelets, they are going to make up less than 1% of the blood volume. We're now going to look at the hematocrit. From the previous slide, we have actually discussed that when we put a blood sample into a glass test tube and then spin it, then allow it to settle, what happens is that the heavy red blood cells, the denser red blood cells settle at the bottom, and then the lighter plasma settles on top. In between these two, we have the Buffy coat, the combination of white blood cells and platelets that settle in between them. Supposing this whole blood volume is 5 liters, the percentage of these 5 liters of this total blood volume that is occupied by the red blood cells is known as the hematocrit. What this means is that if the hematocrit is 40%, what this means that 40% of these 5 liters, 40% of this volume of blood are red blood cells. The other percentage will be the plasma and the Buffy coat. Now this hematocrit differs between adult females and adult males. Adult females have got a lower hematocrit in comparison to adult males. Adult females have got a hematocrit of about 38 to 46%, on average 42%, and then adult males have got a hematocrit of about 40 to 54%, on average 47%. Why do we have such a difference? This difference arises because of two reasons. Number one, testosterone levels. Males have got more testosterone in comparison to females. What this testosterone does in males is that it stimulates production of a hormone known as erythropoietin. What does this erythropoietin do? Erythropoietin is a hormone that stimulates red blood cell production. RBC production is under the influence of erythropoietin. So because of that, males will have more red blood cells in comparison to females. The second reason is because for females that are in their reproductive years, during their menstrual cycles, they tend to lose blood. So because of that, their hematocrit is going to be affected, in that it is going to be lower in comparison to that in males. Let us now look at the components of blood in details. So remember, whole blood is about 8% of the body weight, and it makes up 20% of the extracellular fluid, which is the fluid that is present outside the cells. Of this 8%, 55% is the liquid component of blood, which is the plasma, and then 45% are the formed elements, the cells and the cell fragments. Of the 55% of plasma, 91.5% is water and 8.5% are solutes or solids. Out of that 8.5% of solids, 7% are proteins, while 1.5% are other solutes. Proteins that are present in blood include albumins, which make up 54%, globulins, which make up 38%, and fibrinogen, which make up 7% of this 7% there. Other solutes include electrolytes such as sodium, potassium, and chloride. Nutrients include glucose, amino acids, and lipids. Gases include oxygen and carbon dioxide. When you're talking about regulatory substances, these regulatory substances include substances such as enzymes and hormones. Waste products include urea, uric acid, and ammonia. Though there are other waste products that I didn't mention here, but those are some of the examples. When you're talking about formed elements, we have cells such as red blood cells and white blood cells, and we have cell fragments, which are platelets. Remember, platelets are not complete cells, they just break off from a larger cell during production. Out of this 45%, 99% of that 45% of the formed elements are red blood cells, which make up 4.8 to 5.4 million cells per microliter. The other remaining less than or equal to 1% are the white blood cells and the platelets. White blood cells make up about 5,000 to 10,000 cells per microliter, and these white blood cells are of different types. We have neutrophils, lymphocytes, monocytes, eosinophils, and basophils, which we are going to explore in detail in the next video when we're going to look at white blood cells. Platelets, they form about 150,000 to 400,000 cells per microliter. So if you look at the formed elements, the most abundant formed element are the red blood cells, which make up 4.8 to 5.4 million cells per microliter. So this is a picture of a blood smear, and it is showing you the different components of blood, how they look like under a microscope. So when you look at this blood smear, you realize that there is this whitish background there where the cells are suspended. So this is the blood plasma, the liquid component of blood. Then when you look on top here, and when you also look at this one here, that one there, and that one there, those are white blood cells. This one specifically is a neutrophil. The one at the bottom there is the monocyte. The different types of white blood cells are going to be explored later on. Then when you look at this blood smear, you will realize that there is this numerous blood cell that you can see here. This numerous blood cell is the red blood cell. So these are red blood cells, and you can see how abundant or how numerous they are based on what we are talking about. Then these smaller ones that you are seeing there, they are not really cells. They are not complete cells. They're just cell fragments. They bud off or they break down during production, which we're going to look at later on. So these ones are platelets. So the components of blood can further be divided into two: blood plasma and formed elements. Formed elements, white blood cells, platelets, and red blood cells. When you remove the formed elements from blood, the straw-colored liquid that is left behind is known as blood plasma, or simply just plasma. Like I've mentioned before, this blood plasma is made up of 91.5% water, 7% proteins, 1.5% solutes, and I've given examples of different solutes, electrolytes, nutrients, gases, and so on. My main concern here is that we want us to talk about the proteins. There are three important proteins that I want us to talk about. We have albumin, which makes up 54% of that 7%. We have globulins, which makes up 38% of the 7%, and fibrinogen, which makes up 7% of that 7% of proteins. These proteins, known as plasma proteins because they are found in plasma, they are produced by the liver cells, which are also known as the hepatocytes. However, there's a different type of globulin, which is known as immunoglobulin. Immunoglobulin or immunoglobulins, which are antibodies. These antibodies, despite them being plasma proteins, they are not produced by hepatocytes. They are produced by white blood cells. There's a different type of white blood cell that produces immunoglobulins or antibodies, which is a type of globulin. We are going to look at it under the white blood cells. This slide is showing us the components of plasma, or the substances that are present in blood plasma. When you look at the proteins, we have albumins, which are the most numerous proteins. If you remember, it's about 54% of the 7% of the plasma proteins. What is the function of this albumin? To understand this function, you need to understand that at capillary level, if these are our cells, the space in between these cells is the interstitial space. And if this is our capillary, when blood is passing through this capillary, what happens is that some of the fluid seeps out of this capillary and goes into this interstitial space, the space that is present in between the cells. So what albumin does is that it maintains what is known as osmotic pressure. And this osmotic pressure is the pressure that opposes the movement of this fluid from the blood vessel into the interstitial space, so that just quite enough fluid, or just that the right amount of fluid that is needed should leave the blood vessel and go into the interstitial space. Without it, a lot of fluid would go into the interstitial space, and that can cause things such as edema. So albumin is there to maintain this osmotic pressure that opposes the movement of this fluid from the capillary walls or from the capillary vessels into the interstitial space, the space in between the cells. Then globulins. These globulins, we have different types of globulins. Some of them, such as the immunoglobulins, which are produced by the plasma cells, a type of white blood cell, they help in fighting off infections, and these infections are those that are caused by viruses and bacteria. Then we also have some like alpha and beta globulins that help in transportation of materials such as iron, lipids, and fat-soluble vitamins. For example, vitamin A, D, E, and K. Those are fat-soluble vitamins. They cannot be transported freely in blood. They have to bind to the proteins, and some of the examples of these proteins are these globulins that we have mentioned there. Fibrinogen is very much important in blood clotting. It helps in prevention of excessive blood loss. When the platelets stick together, fibrinogen also goes there and helps in stopping the bleeding. You can think of it like your own bandage system in the body. The other functions of some of these solutes that have been mentioned there, we can take time to look at them, but my main focus here was on plasma proteins and their function. Let us now talk about the formation of blood cells. The formed elements of blood are continuously being replaced. The process by which these blood cells are produced and develop is known as hematopoiesis or hematopoiesis. This process before birth occurs in the yolk sac of the embryo, then later occurs in the liver, spleen, thymus, and lymph nodes. However, the primary site of hematopoiesis changes from the last 3 months before birth, after birth, and throughout life. During these periods of life, the red bone marrow becomes the primary site of hematopoiesis. This process of hematopoiesis begins with a cell that has the ability to differentiate or to become different types of blood cells, and these cells are known as pluripotent stem cells, and they are present within the red bone marrow. This diagram is showing the process of hematopoiesis. The process of hematopoiesis begins with stem cells that have the ability to differentiate into different types of formed elements of blood, and these stem cells are known as pluripotent stem cells. A pluripotent stem cell then differentiates into 2 types of stem cells, the myeloid stem cell and the lymphoid stem cell. The myeloid stem cell then differentiates into 3 progenitor cells, also known as colony-forming units. These 3 colony-forming units differentiate into precursor cells, also known as blast cells. From the first blast cell, we get to produce a red blood cell, also known as an erythrocyte. The process of production of red blood cell is known as erythropoiesis. From the second blast cell, we get to produce platelets, and this process is known as thrombopoiesis. From the third, fourth, fifth, and sixth blast cell, we get to produce different types of white blood cells, such as the eosinophil, the basophil, the neutrophil, and the monocyte. The process of production of white blood cells is known as leukopoiesis. A monocyte can further migrate into a tissue such as a connective tissue and become a macrophage. From this myeloid stem cell also, we can produce another cell that can be found in tissues such as connective tissues, and this cell is known as a mast cell. The lymphoid stem cell gives rise or develops into 3 types of blast cells: the T lymphoblast, the B lymphoblast, and the natural killer cell lymphoblast. The first 2 lymphoblasts become 2 different types of lymphocytes. The first one is the T lymphocyte, also known as the T cell. The other one is the B lymphocyte, also known as the B cell. The B cell can further differentiate into a plasma cell and a memory cell, which we are going to look at in details under white blood cells. The natural killer lymphoblast becomes a natural killer cell, and we are going to look at the functions of each and every white blood cell that we have talked about in the next lecture. This is how hematopoiesis occurs in the red bone marrow. Let us now shift our attention to red blood cells. Red blood cells, also known as erythrocytes, are the most abundant formed elements in blood. These red blood cells possess a very important protein known as hemoglobin, and this hemoglobin is used in the transportation of gases. Hemoglobin carries oxygen from the lungs or the cells, and it also carries about 23% of carbon dioxide, which is a waste product of metabolism from the cells to the lungs. Other features that are unique to red blood cells are that red blood cells do not have a nucleus and other organelles. Examples of organelles that are not present in red blood cells are the mitochondria. So because of that, red blood cells do not perform extensive metabolism such as aerobic metabolism, where it produces ATP in the presence of oxygen because it doesn't have a mitochondria. The other feature is that red blood cells are biconcave discs in terms of their shape. So you can think of this like a donut, but this donut does not have a hole in the middle. The outer part of these red blood cells are thick, while the middle part is flattened and very thin, so it gives it this biconcave disc shape. The other feature is that the red blood cells, in terms of their diameter is about 7 to 8 micrometers. Talking about their cell membrane, the cell membrane of red blood cells is very flexible. By this flexibility, what we mean is that a red blood cell has the ability to squeeze, to change shape and squeeze through narrow blood capillaries. So a blood capillary might be very small and the red blood cell might be bigger, but when it reaches that blood capillary, it is able to deform or to change the shape here and then pass through these small blood capillaries and then be able to go to the other side or to the other parts of the body. If you remember earlier on in the video, I mentioned that the total number of red blood cells is about 4.8 to 5.4 million cells per microliter. This total number of red blood cells differs between an adult male and an adult female, with adult males having more red blood cell count in comparison to adult females, and I've given the reasons in this video to why it is so. So if you have forgotten, just rewind back and you'll be able to find those reasons in this video. So an adult male on average has about 5.4 million red blood cells per microliter, and an adult female has about 4.8 million red blood cells per microliter. Another important concept that I would like you guys to understand is that the structure of red blood cells is highly specialized for oxygen transport. What does that mean is that the structure of red blood cells favors oxygen transportation. And there are three reasons to why the structure of red blood cells favors oxygen transportation. Number one is that all the internal space that is present in the red blood cells is available for oxygen transport. The reason is that there is no nucleus and no organelles that are present inside the red blood cells. So because there's no nucleus and there are no organelles, that space that is present in the red blood cells is just for oxygen transportation. Then the other second reason is that red blood cells do not use up any of the oxygen that they transport. The reason why red blood cells do not use any of the oxygen that they transport is because they lack the mitochondria. They don't have mitochondria. A cell that has the mitochondria has the ability to generate ATP in the presence of oxygen, which is known as aerobic metabolism. So when a cell has got a mitochondria, has the ability to perform aerobic metabolism, they can generate ATP in the presence of oxygen. But because the red blood cell does not have any mitochondria, they cannot use the oxygen that they transport to produce ATP. Then the third reason is that red blood cells have got a greater surface area for diffusion in comparison to other cells that are spherical, to the cells that could be round. For example, red blood cells have got a biconcave disc shape, and that biconcave disc shape increases the surface area for diffusion. The higher the surface area, or the larger the surface area, the greater the rate of diffusion. So gases are going to diffuse faster into and out of the red blood cell. Remember, I mentioned that red blood cells have a protein, hemoglobin. This hemoglobin is very much important in the transportation of gases. Hemoglobin helps in transporting or carrying oxygen from the lungs to the tissues, and it also helps in transporting or carrying about 23% of carbon dioxide from the tissues or from the body cells to the lungs. In terms of structure, hemoglobin is made up of two parts. It is made up of a protein part and a non-protein pigment or a non-protein part. This protein part is known as globin, and the non-protein part is known as a heme. Globin, which is a protein part of hemoglobin, is made up of 4 polypeptide chains. And in an adult hemoglobin, these 4 polypeptide chains are made up of 2 alpha and 2 beta chains. So if you look at this image of hemoglobin here, this light blue chain that is on top there is an alpha polypeptide chain, and this one at the bottom here also is an alpha polypeptide chain. So 2 alpha polypeptide chains. This dark blue chain there and this other dark blue chain at the bottom there, that's the beta polypeptide chains. So we also have 2 beta and 2 alpha, giving us 4 polypeptide chains. The non-protein part, which is heme, is found on these 4 chains. So each polypeptide chain has a heme that is attached to it. So there is a heme here attached to this alpha polypeptide chain on top. There is this heme here attached to this alpha polypeptide chain at the bottom. There is this heme also attached to this beta chain on top. There is this heme also attached to this beta chain at the bottom. If you look at the structure of heme, it looks something like this. Forget about these structures, the CH2s and CH3s. I would like you to concentrate at the center of this heme. At the center of this heme, there is this reddish part that is you are seeing there. That is an iron ion. So every heme has an iron ion at the center. So because we have got 4 heme molecules that are attached to 4 polypeptide chains of the globin, this means we're going to have 4 iron ions in each hemoglobin molecule. So every hemoglobin molecule has 4 polypeptide chains, 4 heme, 4 iron ions. These iron ions that are present in heme bind with oxygen reversibly. So meaning the oxygen at some point has to come out of this heme so that it goes to the cells. So because we have got 4 iron ions in each hemoglobin will transport 4 oxygen molecules, because one oxygen molecule binds to one heme, another one to another. So we have got 4, we are going to have 4 oxygen molecules. So these are the concepts that are very much important that I wanted you to understand under hemoglobin in terms of its structure and how it carries oxygen molecules. If you remember, we talked about hematopoiesis. Hematopoiesis is the process by which formed elements, which are the red blood cells, the platelets, and white blood cells, are produced in the red bone marrow. If we are being specific to the red blood cells, production of red blood cells in the red bone marrow is known as erythropoiesis. We're going to look at erythropoiesis in details in the next slide. For now, I would like you to understand that the cell that is produced at the end of erythropoiesis in the red bone marrow is an immature red blood cell, which is known as a reticulocyte. If there are many red blood cells that are immature, we call them reticulocytes. Once these reticulocytes have been produced in the red bone marrow, they are going to enter the circulation, they're going to enter the bloodstream, and after about 1 to 2 days, they become mature red blood cells. They have the ability now to transport oxygen, and they have the features of a mature red blood cell. A mature red blood cell does not have a nucleus, and also does not have the organelles. If you look at a reticulocyte in comparison to a mature red blood cell, reticulocytes do not have the nucleus. At this stage, when a reticulocyte has been produced, it gets to remove the nucleus. It is that process of the removal of the nucleus by the reticulocyte that gives the red blood cell its biconcave disc shape. But even if the reticulocyte in comparison to a mature red blood cell does not have a nucleus, the reticulocyte has organelles, and it has organelles such as the mitochondria, it has some ribosome, and it has the endoplasmic reticulum. So after 1 to 2 days, that's when these organelles are also removed, and then it becomes a mature red blood cell. This process of erythropoiesis is under regulation of a hormone that is released by the kidneys in response to low oxygen concentration going to the tissues. If there is low oxygen concentration going to the tissues, that low oxygen concentration is what is known as hypoxia. So in response to hypoxia, the kidneys get to release a hormone which is known as erythropoietin, and this erythropoietin stimulates the process of erythropoiesis. It makes the hematopoietic stem cells, the pluripotent stem cells, to differentiate and become erythrocytes. They become red blood cells. So it stimulates the process of erythropoiesis. We're going to look at erythropoietin in just slides from now. So this picture is the same picture that we used when we were discussing the process of hematopoiesis. But our focus here is the process of erythropoiesis, which is the production of red blood cells in the red bone marrow. The process of erythropoiesis begins with a pluripotent stem cell becoming the myeloid stem cell. This myeloid stem cell becomes the colony-forming unit erythrocyte. This colony-forming unit erythrocytes then becomes the proerythroblast, which is the precursor cell. This precursor cell undergoes several divisions, or it divides many times, and then it forms cells that have the ability to produce hemoglobin. They have the ability to synthesize hemoglobin until we get to produce a reticulocyte, which is an immature red blood cell. If you remember what we have just discussed in the previous slide, we said the reticulocyte is the one that gets to remove the nucleus. It ejects the nucleus from the cell, and this process of removal of the nucleus is what forms the biconcave disc shape that we see in a red blood cell. But at this point, this reticulocyte still has some organelles such as the mitochondria, the ribosomes, and the endoplasmic reticulum. Then this reticulocyte gets to enter the circulation. It gets to enter the bloodstream, and after about 1 to 2 days, it matures and becomes a mature red blood cell or an erythrocyte. So this is how erythropoiesis occurs. So when I was introducing erythropoiesis in the previous slides, I briefly talked about the hormone erythropoietin. This hormone erythropoietin is released by the kidneys in response to low oxygen concentration, and this is known as hypoxia. What you have to understand is that the process of red blood cell production, which is erythropoiesis, is under negative feedback control. Negative feedback control. So what happens is that when the blood that is going to the kidneys has got low oxygen, when there is hypoxia, these low oxygen levels in blood is detected by the kidney cells, and these kidney cells, they are going to increase the release of erythropoietin into the blood. What erythropoietin will do is that it is going to act on the red bone marrow and make them produce more red blood cells. It's going to make them release reticulocytes more quickly into the circulation, and that is going to increase the number of red blood cells in the circulation. What this will do is that it is going to increase the oxygen levels. That's why this is a negative feedback mechanism, because what stimulated this whole process are the low levels of oxygen. But after the red blood cells have been produced, we get to have high levels of oxygen now, because we have got more red blood cells that can transmit or that can transport oxygen in blood, and therefore we're going to have a high level of oxygen reaching the tissues. So the whole point here is that red blood cell production is under the influence of erythropoietin, which is secreted from the kidneys in response to low oxygen concentration. After erythropoietin has been released, there's going to be an increase in the red blood cell production, and that is going to cause an increase in the oxygen levels in blood. Now that we have talked about erythropoiesis, we can now talk about the breakdown of red blood cells. Remember, at some point I mentioned that formed elements are continuously being replaced in blood. So red blood cells can only live up to about 120 days. So after 120 days, those old red blood cells are going to be broken down. They're going to be removed from the circulation by the spleen as well as the liver. So the breakdown products, whatever remains from the breakdown or from the degradation of red blood cells, those components that remain are going to be recycled, and they're also going to be reused. Some will be reused in other metabolic pathways. Some are going to be reused in the synthesis of red blood cells. So in the next slide, we're going to look into details how red blood cells are broken down, what components are produced, and how are those components reused or recycled. This diagram is showing the process of both erythropoiesis as well as red blood cell breakdown. So the right side of this diagram is showing the process of erythropoiesis, where the red blood cells are going to be produced in the red bone marrow, and after they have been produced in the red bone marrow, these red blood cells are taken into the circulation, where they're going to stay for about 120 days. Then they become old, their cell membrane has been worn out, and then some of these red blood cells even rupture. And then as the blood is passing through the spleen, through the liver, these red blood cells are going to be phagocytosed by the macrophages. They are going to be eaten up by the macrophages that are present in the spleen, liver, and the bone marrow. What these macrophages, which are the type of white blood cells, will do is that they'll break down these red blood cells. They'll break down the hemoglobin into the globin as well as the heme. The globin, which is a protein part, will further be broken down to individual amino acids. If you remember, the globin is a protein part of the hemoglobin. And this protein part of the hemoglobin is made up of amino acids. So it is going to be broken down to amino acids that are going to be reused for protein synthesis. They are going to be used to produce new proteins. Then this heme, its iron that is present at the center, is in the form of Fe2+, which is the ferrous state. It's a ferrous iron. This iron is going to be converted to the Fe3+ state, which is the ferric iron, and it is going to be taken into the bloodstream, and in the bloodstream, for it to be transferred, it is going to be transferred whilst attached to a protein, and this protein is known as transferrin. So transferrin is a protein that transports the iron in the bloodstream, taking it to places such as the liver, storage sites such as the liver, as well as the muscles. In the liver and other storage sites such as the muscles, this iron is going to be stored in the form of ferritin. So ferritin is another protein which is a storage protein for iron. Transferrin transports the iron, ferritin stores it. When this iron is needed for erythropoiesis, what will happen is that this ferritin is going to release this iron, and this iron is going to bind to transferrin, transported in the bloodstream, and then it goes to the red bone marrow for erythropoiesis in order to produce more red blood cells. Now the other part that remains for the heme is going to be converted to biliverdin. Biliverdin is a greenish substance that is produced when the remaining part of the heme has been metabolized or has been broken down. Then this biliverdin is going to be converted to a yellow-orange substance which is known as bilirubin. This bilirubin is going to be taken to the liver, and in the liver this bilirubin is going to be excreted, or it is going to be added to the bile, and this bile is going to go into the small intestine. In the intestines, this bilirubin is going to be acted upon by the bacteria that is present in the intestines, converting it to urobilinogen. Urobilinogen has got two pathways. Some of it is going to go back into the circulation, and that blood, as it is passing through the kidneys, that urobilinogen is going to be converted to urobilin, and then this urobilin is going to be excreted in the urine, giving the color of the urine, making it one that makes the color of the urine. Then some of this urobilinogen also, in fact most of it, is going to be converted to stecobilin, and this stecobilin is going to be excreted in the feces, and it gives the color to the feces. So this is how red blood cell breakdown happens. In summary, the globin is broken down to amino acids which are used for protein synthesis. The heme has the iron which is removed, and then it is stored. When it is needed for erythropoiesis, it is taken back to the red bone marrow. The remaining part of the heme converted to bilirubin, and this bilirubin can be excreted in the kidneys, in the urine as urobilin, and then it can be excreted in the feces as stecobilin. We can now shift our attention to blood groups and blood types. You have to understand that individuals have different blood groups, and these blood groups are determined by the presence or absence of what are known as antigens or agglutinogens. Where do we find these antigens or agglutinogens? They are found on the cell membrane of a red blood cell. So if this is our red blood cell, on their cell membrane you are going to find what are known as antigens, and these antigens can either be a glycoprotein or a glycolipid. What is a glycoprotein? A glycoprotein is a protein that has a carbohydrate attached to it. Is a protein that has a carbohydrate attached to it. Now, there are two types of blood group systems that we have to look at. We have to look at the ABO system and the rhesus system. In the ABO system, there are two types of antigens that can be present on the red blood cell of an individual. We can either have antigen A or antigen B. So, if an individual's red blood cells have antigen A, then that person is going to be blood group A. If an individual's red blood cell has antigen B on the surface of the red blood cell, then that individual is going to be blood group B. Individuals can also have red blood cells that have both antigen A and antigen B, and such type of a blood group is known as blood group AB. So these are the antigens that we are talking about. In some circumstances or in some individuals, their red blood cells do not contain any of these antigens. They don't have any of these antigens. They don't have antigen A. They don't have antigen B. Such type of a blood group is known as blood group O. Apart from antigen A and antigen B, in the rhesus system, an individual's red blood cell can have another antigen apart from antigen A or antigen B. They can have another antigen which is known as the rhesus antigen. And if an individual's red blood cell have a rhesus antigen, their blood group is going to be designated, or it is going to be known as a rhesus positive blood group. But if that individual's red blood cells do not have the rhesus antigen, then that individual's blood group is going to be designated, or it is going to be referred to as rhesus negative blood group. So when we are discussing blood groups, we combine both the ABO system and the rhesus system. So if an individual has antigen A and they also have rhesus antigen, that individual's blood group is going to be referred to as blood group A positive. If an individual has antigen A but they don't have the rhesus antigen, that individual's blood group is going to be known as A negative because there's no rhesus antigen. If an individual has antigen A as well as antigen B, that individual has blood group B, but they have the rhesus antigen, then that individual's blood group is going to be known as AB positive. When the rhesus antigen is negative, then that individual is going to be AB minus. So this is the same for blood group B. They have the rhesus antigen B positive. They don't have the antigen B negative. Same with blood group O. They have the rhesus antigen O positive. They don't have the rhesus antigen O negative. So that's what you have to understand about the ABO system and the rhesus system. The other important thing that you have to understand about these blood groups is that in the plasma of an individual, you also have what are known as antibodies. These are proteins that we talked about when we were talking about immunoglobulins. When we talked about the globulins, and I said there's a type of globulins which is known as immunoglobulins or antibodies. They are found in the plasma. So these antibodies are also known as agglutinins. What these antibodies do is that they can recognize and bind or attach to the antigens that are present on the red blood cells, and then they can cause these red blood cells to clump together. They can bind several red blood cells, causing them to clump, and then these red blood cells are not going to be able to transport oxygen. We're going to have some reactions, and this mostly tend to happen when an individual has been given the wrong blood that has got a different blood group to theirs. So the reason why I'm talking about these antibodies is that the plasma of an individual cannot have the antibodies against their own red blood cells, but they'll have antibodies against another person's red blood cells. What do I mean by that? Before we talk about that, you need to understand that there are two types of antibodies that an individual's plasma can have. They can either have antibody A or they can have antibody B. So, supposing we have an individual who is blood group A. This individual who is blood group A cannot have antibody A in their plasma, but they are going to have antibody B. An individual who has got blood group B cannot have antibody B in their plasma, but they will have antibody A. So an individual cannot have antibodies against their own red blood cells. So if you are B, you have blood group B, you cannot have antibody B because your red blood cells have antigen B. And supposing you are to have antibody B, this antibody can bind these antigens present on your red blood cells, clamp them together, and then avoid them from performing their function. We're going to discuss more about antibodies in the next slide. So this picture is showing what I've already explained, and I've mentioned that in the ABO system, if an individual's red blood cell has antigen A or it has antigen B, that individual will have blood group A and they'll have blood group B respectively. If they have got both antigen A and antigen B, that individual is going to be blood group AB. But if they don't have either antigen A nor antigen B, then that person is going to be blood group O. However, when it comes to the plasma, where you find antibodies, the rule is that an individual shouldn't have an antibody against their own red blood cell. So if that person has antigen A, you shouldn't expect them to have antibody A in their plasma. So for an antigen A red blood cell, the plasma should have anti-B antibody. For a B antigen red blood cell, the plasma should have anti-A antibody, not anti-B antibody. They shouldn't have an antibody against their own red blood cell. But because blood group AB has both antigen A and antigen B, then their plasma should not have both antibody A and antibody B. That's why you find that in the plasma of an individual who has who have got both A and B antigen, they are not going to have any antibody in their plasma. But if you look at blood group O, because their red blood cell does not have antigen A nor antigen B, their plasma has both antibody A and antibody B. So it's important that you understand that, because it helps in understanding how transfusion of blood happens. On this diagram, I'll briefly just talk about how transfusion happens. So you need to understand that when you're talking about blood group A, if that blood group A is A positive and another one is A negative, this A negative can only receive blood from two types of donors. One who is A negative can give to A negative, and one who is O negative can give to O negative. O is the universal donor. They can give to anyone, regardless of their blood group. But you also have to put in mind the rhesus factor, that a rhesus factor negative donor can give to a rhesus factor negative recipient and also to a rhesus factor positive recipient. But a rhesus factor positive donor cannot give blood to a rhesus negative recipient. They can only give blood to a rhesus positive recipient. So that's why you have not seen me adding A positive here giving to A negative, because an A positive should just give to an A positive, but an A negative can give to an A positive, then an O positive can also give here, and an O negative can give to this one there. So you have to understand those principles. Because I've explained those principles, you can try them out, but my main focus here is to just make you understand which blood group is giving to which blood group is receiving from which blood group. So A can receive from A and O, but cannot receive from B and AB. B can receive from B and O, but cannot receive from A and B. Then when you're talking about AB, AB is a universal recipient. It's a universal recipient, meaning it can receive from all types of blood group. It can receive from A, it can receive from B, it can receive from AB, it can also receive from O, but it cannot give to any of those. It just gives it to itself. So it's a universal recipient. O is a universal donor. It can give to A, it can give to B, it can give to AB, but it cannot receive from these. So you can see this is an incompatible donor blood type. I've just used it to explain that it cannot receive from A, it cannot receive from B, it cannot receive from AB. Okay, but it can give to all of these but not receive from them. It just has to receive from O itself. So those are the principles that guide blood transfusion in simple details. So we have come to the end of this lecture. I'll see you again in the next lecture, where we're going to discuss the white blood cell now. Because we are done with the red blood cell, we're going to discuss the white blood cell. Then after that, we'll discuss platelets. So please make sure you subscribe, make sure you like, make sure you share, make sure you turn on the notification button so that every time I post a new video, you're going to be notified. Thank you, and see you in the next video.